Hello, we're glad you've joined us for this live webinar today, Mapping Nuclear Exosome-Targeted poly-A tails with 3' end RNA sequencing. I am Brenda Kelly-Kim of Labroots, and I'll be moderating this session today. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. It's brought to you by Lexogen. Lexogen is a transcriptomics and next-generation sequencing company focusing on development of products enabling complete transcriptome sequencing. Lexogen was founded in 2007. It is based in Vienna, Austria, and has a subsidiary in New Hampshire, United States. To learn more, please visit us at www.lexogen.com. Now let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience any technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left. I'd now like to present today's speaker, Kevin Roy. Kevin obtained his PhD in molecular biology in the laboratory of Dr. Guillaume Chanfrot at UCLA. There, he studied environment-dependent RNA degradation pathways in yeast and developed high-throughput methods to map poly-A plus 3' ends of short-lived RNA species. He has a strong background in yeast genetics and molecular biology, as well as in high-throughput sequencing techniques and data analysis. He is currently a postdoctoral scholar in the laboratory of Dr. Lars Steinmetz in the Department of Genetics at Stanford University. His current interests include high-throughput systems genetics with a focus on how genetic variation interacts with environment to produce phenotype. Dr. Roy will now begin his presentation.
Hello, everybody. Good morning to those of you on the West Coast, and afternoon and evening to those of you out on the East Coast and in Europe. My name is Kevin Roy, and I am currently a postdoctoral researcher at Stanford. Today I'd like to talk first about RNA sequencing in general, and then focus on specifically 3' end RNA sequencing and the benefits that it exhibits for certain studies. And then I'd like to go into some of the biology that we uncovered by looking specifically at the nuclear exosome-targeted RNAs. So first, if we look at what RNA sequencing can show us, is it really reveals what portions of the genome are expressed. So in particular, whole transcript RNA sequencing is a technique in which the entire transcript is fragmented, and then the regions are sequenced individually. And this enables us to understand the transcript structure, so we can infer the start site of the gene, the end site, as well as where the transcript is spliced together. Another important benefit of RNA sequencing is that it allows us to quantify the expression level of genes across the genome, so we can understand how genes are expressed and how they are regulated. But whole transcript RNA sequencing requires that many reads be assembled across the gene in order to map its structure. And so this allows us to understand these aspects of how the transcript is formed, but in some cases we might be interested in 3' end sequencing. And so why would we want to perform 3' end sequencing as opposed to the whole transcript sequencing? Well, 3' end sequencing is cost effective and it's efficient because it counts only the ends of the transcripts, and only one cDNA is generated per transcript. In addition, the differential expression analysis, so determining how the expression level of different transcripts in a sample has changed, is simpler for 3' end sequencing because it's not complicated by the length of the transcript and the variation in detection across the transcript. Furthermore, 3' sequencing allows for the precise mapping of cleavage and polyadenylation sites, giving insights into the mechanisms that generate the alternative 3' untranslated region selection. So for this particular gene, YNL067W, you can see how the traditional RNA-Seq and the 3' end RNA-Seq provide different advantages for understanding the transcript. So the traditional RNA-Seq actually does a fairly poor job of capturing the ends of the transcript. And that's a consequence of how these libraries are prepared. The 3' end sequencing, however, only focuses on the 3' end and is very efficient at detecting exactly where these transcripts end. So you can see that with 3' end sequencing, there is one major polyadenylation site, but there are also a few other polyA sites in this transcript that would not be well identified with the traditional RNA-Seq method. So if we look at how 3' end RNA sequencing is accomplished, it's a fairly simple protocol, and another advantage over the whole transcript sequencing is that the library preparation is quite efficient, quite simple. So the first step involves the oligo-dt binding to the poly-A tail that is present on messenger RNAs. And this allows for the reverse transcriptase to generate a single-stranded DNA from the RNA. And from there, the library preparation is conducted with random priming of second-strand synthesis and then PCR amplification with barcodes that allow for multiplex high-throughput sequencing. And so when we first started to perform 3' end sequencing on wild-type yeast, we were able to get a large number of reads using the QuantSeq platform, and we obtained over 10 million reads for one sample in this case. And we noticed that we only roughly half of the reads mapped to unique positions within the genome, and nearly 40% of the reads were mapping to multiple positions. And when we looked at the FASTQ-C report on the overrepresented sequences in this sequencing experiment, we noticed that many of these sequences mapped to the ribosomal RNA. And so in this particular case, you're seeing the ribosomal RNA, the gene encoding ribosomal RNA, which is encoded by multiple copies in the genome. And a comparison of the whole transcript sequencing, which results in background due to this abundant transcript, with the 3' end sequencing reveals a hotspot that is specific to 3' end sequencing. And this is a contaminant in the data set, and this actually corresponded to 30% of all the reads in our data set. So if we zoom in on this hotspot, we can see that the oligo-dt was likely priming at these short stretches of imperfect A's that typically had a G or two in the middle. And so from looking at the types of mispriming that we were getting from this abundant RNA, we're able to determine that oligo-dt can efficiently prime under the conditions of our library prep imperfect stretch of 6 A's that can be interrupted with up to 2 G's. And the GT base pair can be, it's a wobble pair, and that explained why we were getting some of these contaminant reads. So when we look back at the protocol, we noticed that one of the critical steps is the hybridization of the oligo-dt primer to the poly-A tail. And so a major factor in how the priming will occur is the temperature of the hybridization and the reverse transcription. So what we did for this step was we decided to raise the temperature from 37 degrees to 42 degrees, and perhaps more importantly, we keep the samples on the heating block during this entire step. And in addition, one other modification we made was to increase the cycles from 12 to 15 cycles. And so now, by these slight modifications, we were able to obtain a huge increase in the quality of the library and decrease the internal priming. So here you can see the results of the sequencing library at 42 degrees with the reverse transcription step versus the 37 degree step. And so now instead of 55% of the reads mapping uniquely, we now have over 90%. And this corresponded to a more than tenfold, a roughly tenfold reduction in the internal priming. And so now we notice the overrepresented sequences were much lower in abundance. And in this case, there was one sequence that popped out to us, which was a large stretch of T's. And so this particular artifact, we're not sure exactly the source of it, but it may be due to slight contamination with a read 1 primer that does not contain the poly-ATN. So based on all of these artifacts, we developed a computational filter that would improve the quality of the 3' ends that we identify. And so the first filter utilizes the 6 base pairs downstream of a poly-A site, and it looks for whether or not there are A's and G's. And if there are more than 4 A's and no C or T, so in other words, up to 2 G's and 4 A's, then this is likely an artifact, and so we screened those sites out. Another filter looked at the 18 base pairs downstream of the poly-A site. And if this stretch of 18 nucleotides contain greater than 12 A's or equal to 12 A's, this was also likely to be a mispriming event, and so we screened these out as well. And due to the poly-T reads that we identified, another filter we employed was to look at the 18 base pairs upstream of poly-A sites, and we would filter these poly-A sites if there were more than 15 or more A's in this window as well. So after employing these 3 filters, if we look at the reads from these libraries, the reads mapping to non-open reading frame regions, and so these are reads that are mostly in the 3' UTR region where we expect, the filter that we employ filters out roughly 700,000 out of the 5 million reads. And the most of the reads are filtered with this downstream 6 base pair AG filter. The other 2 filters remove roughly 10% of that, 60,000 reads. So if we look at the reads that we're actually mapping to within open reading frame regions, we notice that many of these reads were likely internal mispriming events. And so this filter actually removes almost half of these reads, so 66,000 are removed, and the remaining 110,000 are could represent real internal polyadenylation events, and then the other filter removes only 6,000. So in total, using these computational filters in combination with the modified library preparation protocol resulted in a higher confidence calling of poly-A sites, and 98% of the poly-A sites identified were not in open reading frame regions, which is consistent with what we know about the polyadenylation in the cell. So here's an example of that ribosomal RNA hotspot, where if we look at all the poly-A sites, this actually corresponds to 30% of the entire reads in the sequencing library were mapped to just this small region of less than 50 base pairs. And if we look at the computational filter, it was able to remove the majority of these reads. Now this is the library prepared with 37-degree reverse transcription. When we increased the reverse transcription to 42 degrees, this contamination to begin with was much less, so 3% of all the reads. And the filter, if you look at the passing poly-A site filter, it removed the majority of these. So we think that with these 2 steps, improving the protocol with a higher temperature of reverse transcription and computationally removing likely artifacts from the poly-A sites, we are improving the confidence in 3' end calling. So now we wanted to get to some of the biology, and our lab is interested in the RNA processing of transcripts, and in particular, the different types of RNA processing that exist in the cell. So the ends of genes are mostly determined by the RNA processing signals. So the 5' end of a gene is where the transcription begins, and the 3' end of a gene is usually not where the transcription ends, but it's where the transcript is actually cut or processed. And much of the RNA in the cell is stabilized and exported, but a large fraction is also degraded. And we're interested in the pathways that that make this choice of either stabilizing or degrading the RNA. So the cleavage and polyadenylation machinery is the primary mechanism for stabilizing RNA, and this is used on the protein coding genes to make messenger RNAs that are ready for translation. However, a large fraction of the transcription in the cell is actually degraded right away. And so these transcripts, one of the major mechanisms to degrade these transcripts uses the Nrd1-Nab3 RNA binding proteins, and these are RNA binding proteins that recognize short sequences, and they result in the degradation of the transcript. And then there are a couple of other mechanisms that also degrade transcripts. One of them is the RNase 3 endonuclease. Another one that was recently uncovered is physical roadblocks in the DNA. So DNA binding proteins that just block RNA polymerase progression also lead to the degradation of the transcript. So what we noticed was that all of these different non-canonical termination pathways, they lead to the degradation of the transcript through a common pathway that involves first polyadenylation by a polyA polymerase different from the canonical polyA polymerase. That polyA polymerase is called TRF4, and it is a part of the TRAMP complex. And that is normally in wild-type cells followed by rapid degradation by the nuclear exosome. And the Rrp6 subunit is important for initiating that degradation. So if we look at the detailed mechanism involves the addition of an oligo-A tail, and this oligo-A tail is then transferred through the nuclear exosome like a thread, and the core part of the nuclear exosome accommodates this thread, and the Dis3 subunit at the bottom of the exosome catalyzes the complete degradation of these transcripts. So what we reasoned was that if we can inactivate the nuclear exosome, we will stabilize these transcripts, and we can then look at the 3' end sequencing of these unstable transcripts, and by comparing these to wild-type cells, we can gain a good picture of how the transcripts are degraded by looking at their 3' ends. And so when we first started this project, there were a couple of options for RNA sequencing of the 3' ends, and one of them that was attractive was the direct RNA sequencing, which conceptually is very simple. It just involves the single molecule sequencing of RNAs directly from their natural polyA tails. The other method is the QuantSeq 3' end library prep method, which I just spoke about. And if we look at the pros and cons of these, the pros of the direct RNA sequencing are that there are no sample manipulation or library prep involved. There's no reverse transcription or amplification bias. However, the pros for the QuantSeq method is that it's incredibly cost-effective in comparison, and multiplexing is possible, and because it's utilizing the Illumina sequencing platform, long reads of 50 or even 100 nucleotides are possible, and there are few errors in comparison. And the cons of the direct RNA sequencing is that the multiplexing is not possible. It's in comparison to QuantSeq, an expensive and slow method, and the short reads of 20 to 25 nucleotides are also error-prone. The QuantSeq method can have internal priming and AG-rich sequences, and as it involves PCR amplification, there is the potential for amplification bias, and also the library size selection can introduce bias, but these steps can all be mitigated. So if we look at the direct RNA sequencing results and what they revealed for these samples, when we inactivated the nuclear exosome, we can see that a substantial fraction of the transcripts map to the 3' UTR, which is what we expect for the protein coding mRNAs. However, we see that in these cells, a large fraction of the transcripts come from these other categories of genes, in particular the SNO RNA genes, which are small nucleolar RNAs that are involved in ribosome biogenesis. And we also see these other cryptic transcripts, in green, the cryptic unstable transcripts, and these other categories of transcripts that have ill-defined function but that are pervasively transcribed by the RNA polymerase. In comparison, wild-type cells exhibit nearly 90% of the reads mapping to the 3' UTR. So we figured that this was an effective approach for understanding the nuclear exosome targets, and we wanted to compare it to the QuantSeq method. So here's a comparison of the exact same RNA samples sequenced on the QuantSeq platform. And you can see that we get a larger fraction of reads mapping to the 3' UTRs. And the relative ratios of the other categories are similar. And so we're not quite sure of the reason for this discrepancy in the percentages of percentages that we see in the different categories. But we think that it may have to do with the higher error rate and the lower read length, the shorter read length of the direct RNA sequencing, resulting in 3' UTR regions not mapping as well. So if we look at the SNO RNAs, these small nucleolar RNAs, which as a class of genes represented a large fraction of the cellular poly A plus RNA after nuclear exosome inactivation, we can see as a class with the meta-analysis 2 prominent peaks. And so what meta-analysis does here is that it takes individually each of the 77 SNO RNA genes, and what we're doing is normalizing the total sequencing signal in the region of these genes, and after normalizing to the same arbitrary constant, we can sum them up. And by looking at them all together, we can see trends that we might miss by looking at any individual SNO RNA. And so here we can see that the two methods, direct RNA sequencing and the QuantSeq sequencing, actually give pretty good agreement in the relative location of these peaks. So we can see one peak right at the mature 3' end of these SNO RNAs, and we can see another peak 25 nucleotides downstream. After that, we see this broad range of signal. And that broad range of signal we think is, we initially suspected was the actual termination product from the NNS pathway, the Nrd1-Nab3 termination pathway. And the 25 nucleotide length peak, we noticed matched the length that is accompanied by the nuclear exosome channel. And so we suspected that perhaps what we're seeing here is a processing intermediate, so that we haven't fully inactivated the nuclear exosome. In these previous experiments, we deleted the Rrp6 subunit at the top here in red. And however, in the absence of Rrp6, the nuclear exosome still has some function. And so in order to fully inactivate it, we also had to get rid of the Rrp44, or otherwise known as Dis3 function. However, Dis3 is an essential gene, and so we couldn't simply delete it. So what we decided to do was we used a conditional depletion of Dis3 with the anchor away method. So the anchor away method is a very effective method in yeast to rapidly deplete nuclear proteins and move them into the cytoplasm. And the reason it's so effective is that it involves a very abundant ribosomal protein, RPL13A, that is modified with a tag called FKBP12. And in the absence of rapamycin, its ligand, there's no interaction between RPL13A, the FKBP12 subunit of RPL13A, and the protein of interest. However, the protein of interest, in the presence of rapamycin, its tag with the FRB binding domain rapidly forms a complex with the FKBP12, and this is a very stable complex that results in the export of this protein into the cytoplasm. And so this method has been shown before, and we thought with the QuantSeq method we could multiplex many samples using mutants that are modified with this method. And so we conditionally depleted the Dis3 catalytic subunit of the nuclear exosome in addition to the Rrp6 subunit. So what you can see here is that the depletion of Rrp6 alone resembles the results that we obtained with the deletion of this gene. But if we deplete both Rrp6 and the Dis3 subunit, we get a larger fraction of reads that are not mapping to 3' UTRs, but instead are mapping to these other classes of transcripts that are known to be targeted by the nuclear exosome. So we went forward with this approach, and what we did was we looked at the total nuclear exosome targeted RNAs, and we wanted to determine where they were mapping to, and what types of genes are targeted by these by the nuclear exosome. So what we did was we clustered these poly-A sites, and we chose to cluster poly-A sites that were within 20 base pairs of one another. And these clusters would then, when they were separated by greater than 20 base pairs, were called as different clusters. And so we chose a cutoff of 2 reads per million for the clusters, for the cluster peaks. And so when we looked at where these clusters were mapping, we noticed that most of them were mapping in 3' UTR and intergenic regions. And when we fully inactivated the nuclear exosome by depleting both subunits, the intergenic region clusters were the regions as well that were previously known to be targeted by the nuclear exosome. So the fact that we identified clusters in intergenic regions, in regions that weren't previously known to have transcripts, led us to believe that the QuantSeq method was giving us unprecedented resolution in identifying the transcription that leads to degradation. So we first looked at the combined depletion of both of these subunits of the nuclear exosome, and what we noticed was that this 25 base pair peak, so if we look downstream of the SNO RNAs, in the Rrp6 depletion alone, we can see that the peak is predominant along with the peak at the mature 3' end. When we remove both subunits of the nuclear exosome, we can see that the plus 25 base pair peak is substantially reduced. And so this led us to believe that we were effectively inactivating the nuclear exosome. And these data led us to propose the following pathway for the 3' end maturation of SNO RNAs. And so the transcription termination depends on the NNS pathway, the Nrd1-Nab3 RNA binding proteins, which recruit the Sen1 helicase. This leads to the release of the transcript and termination of transcription. What happens first is that a short poly-A tail is added to these precursors. This poly-A tail is then degraded by the nuclear exosome in a specific mode of degradation that involves the Dis3 exonuclease. However, this exonuclease is paused when it reaches plus 25 base pairs because the central channel of the exosome is physically blocked by the 3' end structure of these RNAs. And after that, the nuclear exosome disengages, and another poly-A tail is added by the TRAMP complex, which is then fully degraded by the Rrp6 subunit of the nuclear exosome. So in order to fully inactivate the nuclear exosome, depleting both of these subunits was really the most important strategy. And so the next thing we wanted to determine was if Oligo-dt is actually priming these non-canonical poly-A tails efficiently so that we are obtaining an unbiased view of these transcripts. We were concerned that if the tails were too short, perhaps we're only seeing some of these non-canonical targets and not the full picture. So normally in wild-type cells, the canonical mRNA poly-A tails are quite long in comparison. They're 30 to 80 nucleotides long in yeast. The non-canonical poly-A tails are quite short in comparison. So these can contain between 1 and 12 A's. And we knew from previous work that after inactivating the nuclear exosome, the TRAMP poly-A complex actually continues to add short stretches of A's at a time, and so the A tail can get lengthened. But we weren't sure if this was universally occurring on all transcripts. So what we did was we decided to employ an in vitro poly-adenylation treatment. So we took purified RNAs from the cell, from either the wild type cells or the cells in which we inactivated the nuclear exosome. And you can see with the question mark, we weren't 100% sure if the nuclear exosome targeted RNAs were obtaining long enough poly-A tails for us to see all of them. And so what we did to answer this question was we treated these RNAs with ATP and E. coli poly-A polymerase, and this added universally long poly-A tails of over 100 A's. And so we added poly-A tails to mature RNAs that normally don't contain them. And we also added poly-A tails to the nuclear exosome targeted RNAs to make sure that we were detecting all of them. In addition to that, mRNAs also were lengthened by this procedure. And so first, if we look at the signal downstream of the SNO RNAs, when we inactivate the nuclear exosome, we see a number of peaks that seem to have the highest level of signal in the 200 to 300 base pair region downstream of the SNO RNAs. And what we did was, after we in vitro polyadenylated all of the RNA, we see that now by far the most predominant signal is the mature 3' end of these SNO RNAs, and that's because most of the SNO RNA in the cell is mature, fully processed SNO RNA. But if we block out that signal and instead look at the nuclear exosome targeted polyAs, we can see that the in vitro polyadenylation treatment reveals a remarkably similar profile of these unprocessed 3' ends. And so from these data, we could conclude that what the QuantSeq library preparation protocol is efficiently priming these nuclear exosome targeted poly-A tails. And so the next question we had to get into more of the biology was, are these poly-A tail transcripts that we identify upon nuclear exosome inactivation really representative of the true sites of termination of RNA polymerase by the NNS pathway? And so what we decided to do was look at previous data in combination with data that we were obtaining. So at first, if we look at the signal upon the depletion of Rrp6, we can see the mature poly-A tail peak, the plus 25 nucleotide peak, and the peaks that we're really interested in are the ones downstream that are representative of the termination site. So if we inactivate the termination mechanism in combination with the nuclear exosome inactivation, we can do that by depleting the Sen1 subunit as well. So when we deplete the Sen1 subunit, we see a loss of these termination peaks. However, the mature peak and the plus 25 nucleotide peak do not go away, and that confirms that these indeed are not termination sites, but rather the processing sites. Now, if we look at previous data that had examined RNA polymerase II occupancy by CHIP in cells that also have the Nrd1 subunit of the NNS pathway inactivated, we can see that the regions where RNA polymerase normally terminates, it agrees well with the regions that we're identifying by the 3' end sequencing method. And the peaks that we identify in our sequencing methods agree well with the inactivation of the NNS pathway and where the RNA polymerase seems to no longer terminate, as you can see in green. We also compared it to other data as well that had looked at RNA polymerase II PAR clip cross-linking to RNAs that are bound to RNA polymerase II in the wild-type cells as well as in cells that have the NNS pathway compromised with either the Sen1 mutation inactivation or the Nrd1 inactivation. And we can see that the Nrd1 inactivation gives a clear inflection point where the RNA polymerase II is no longer terminating and begins to accumulate downstream. The Sen1 inactivation gives a higher level throughout, so it's harder to interpret the termination site from the Sen1 inactivation. So when we looked at other factors that might be involved in termination, we mapped these poly-A tails onto the nucleosome occupancy, and we noticed that when the nucleosome occupancy began to increase downstream of these no-RNAs, the major termination peaks were coinciding with the nucleosome occupancy peaks. However, we noticed that a number of the peaks upstream of that, but downstream of the plus 25 nucleotide peak, seemed to be highly predominant. And when we looked at these, we noticed that these peaks were directly upstream of DNA binding protein. And so we could divide these two peaks depending on the presence or absence of a DNA binding protein in the immediate vicinity downstream. And so you can see by dividing these two classes of peaks, we can clearly see two distinct classes of termination of RNA polymerase II. So on the far right are the peaks where we did not identify a DNA binding protein downstream. We can see a much broader termination region that peaks around 250 base pairs downstream. In the presence of DNA binding proteins, the termination peaks are much sharper. And both of these termination peaks are highly depleted when we remove the Sen1 factor from the nucleus, indicating that these are NNS termination events that we're identifying. And we can then, in the meta-analysis, we can look at the RNA polymerase II occupancy at these peaks, and we also identify that what we're seeing with the nuclear exosome poly-A tails agrees well with the previous regions that have been mapped by Pol II occupancy. But the advantage of the nuclear exosome-targeted poly-A tails is that we get a high-resolution view of where the transcription is actually terminating. And so we also, in comparison with the PAR-CLIP RNA cross-linking data, we were also able to verify that these poly-A tails were mapping well with these data. And so what we could see is that as a class, the non-roadblocked RNAs without the DNA binding proteins downstream appear to be blocked by the nucleosome occupancy, whereas the roadblocked RNAs in the middle panel at the bottom contain these DNA-resistant footprints directly downstream of each peak. And so we could conclude that the NNS termination appears to require not only RNA binding signals, but also obstacles in the DNA, either DNA binding proteins or nucleosomes. And so we wanted to take a step back and look at all of the clusters that we identified with the 3' sequencing. And so here we're looking at all roughly 3,000 clusters, and we're separating them on the basis of whether or not they have a DNase-resistant site. And so when we map the RNA polymerase II occupancy on these two data sets, we see vastly different behaviors. So in the absence of a downstream DNase-resistant site, we see that RNA polymerase II begins to accumulate near the termination site, indicating pausing, and then at the termination site rapidly falls off. In the presence of a downstream DNA binding protein, the accumulation of RNA polymerase is much more pronounced at the termination site. And we can see that when the RNA polymerase is inactivated with the Sen1 or the Nrd1 depletion from the nucleus, the RNA polymerase II occupancy is increased at these termination sites, indicating a lack of termination. So when we look at the non-exosome targeted poly-A tails, so these are the poly-A tails in wild-type cells that normally, that are typically on the protein-coding mRNAs, we can see that this behavior is not observed. And so we can see that there is an accumulation of RNA polymerase in at the ends of these genes at the 3' processing sites, and this is in agreement with previous studies. But we don't see the same behavior. So we can conclude that we've identified that the NNS pathway utilizes these two distinct modes of termination, and the 3' end sequencing allows us to precisely map exactly where the RNA polymerase is terminating in the case of these nuclear exosome targeted poly-A tails. And so what we also noticed was that the same DNA binding proteins that were blocking RNA polymerase were previously shown to block DNA polymerase. And so it suggested that these DNA binding proteins had some sort of special ability that controlled both DNA and RNA synthesis. And so we processed the previously mapped DNA 3' end data, and DNA replication occurs in two directions because of the anti-parallel nature of the double helix. And one of the directions in the lagging strand, called the lagging strand synthesis, involves repeated rounds of DNA synthesis. And the lagging strand synthesis encounters the same roadblocks that RNA polymerase does. And so you can see here, when we map the nuclear exosome targeted poly-A tails alongside the Okazaki fragment 3' ends from the lagging strand synthesis, we can see they map almost directly on top of each other. However, there is a slight difference in the location of the peaks. And so if we look at the TF3B transcription factor, which binds DNA upstream of tRNA genes, we can see a 9 base pair difference in these peaks. And we thought perhaps this is a characteristic signature of these different modes of roadblock, different modes of DNA and RNA synthesis. And so to confirm that, we looked at another DNA binding protein called Rrb1 that also blocks these synthesis modes. And we saw the same distance of 9 base pairs from the nuclear exosome targeted poly-A tail to the Okazaki fragment 3' end. And so the QuantSeq method allowed us to map these precise peaks and also enabled us to utilize many different mutants, which you see here in the colors at the bottom. And by inactivating different genes, we were able to prove that these peaks are indeed roadblocked by the factors that we've identified. So in conclusion, we were able to determine that with a 3' end sequencing, that the termination sites from the NNS pathway have different profiles depending on the presence or absence of a DNA binding protein. So in the absence of a DNA binding protein, the termination occurs over a broad region. And in the presence of a DNA binding protein, there's a sharp peak of termination that occurs. And the location of that termination is defined by the presence of the DNA binding protein. And so in other work that I don't have time to go into, we were able to determine that the level of Nrd1 and Nab3 binding sites in the RNA actually determine whether termination will occur, but that this needs to be taken into context with the DNA binding protein, so that if there are a medium level of these Nrd1 and Nab3 binding sites, in the presence of a DNA binding protein, termination will occur, but in the absence of a DNA binding protein, the termination is actually quite inefficient. And also, interestingly, what we observed is that even in the presence of a DNA binding protein, if the level of these Nrd1 and Nab3 sites is quite low, termination will actually not occur. And so RNA polymerase is actually able to dislodge the downstream protein and remove it from the template. So now I'd like to go over some of the conclusions of this talk. First, discussing the technical aspects of preparing 3' end libraries. We can conclude that QuantSeq does provide an efficient, rapid, and cost-effective method for quantitative analysis of the RNA 3' ends. We found that performing the reverse transcription at 42 degrees as opposed to 37 degrees dramatically reduces the aberrant internal priming that we observed at these imperfect A sequences. And a key point here is that the samples need to be kept on the heating block during the addition of the reverse transcriptase enzyme. We developed a computational filtering method that's consistent with other methods, but we added specific upstream filters as well to remove the artifacts from the poly-T reads and to reduce the false positive results of 3' end identification. And we were able to multiplex nearly 40 samples with up to 20 strains in duplicate and still obtain between 2.5 to 5 million reads per sample. So this allowed us to really do a lot of biology and to uncover how the RNA polymerase termination was occurring. And so we also were able to conclude that the QuantSeq method is capable of mapping nuclear exosome poly-A tails in an unbiased fashion so that the poly-A tails are indeed long enough, and that these reveal the precise locations of the NNS termination pathway. We're also able to discover that it's not just the RNA signals that matter in defining termination, but that the chromatin obstacles, so the DNA binding proteins that are impeding RNA polymerase II, are very important in promoting termination. And lastly, we can conclude that these common genomic elements that bind DNA, they terminate both the DNA synthesis and the lagging strand replication and RNA polymerase II transcription. And so it suggests that the cell is utilizing a common set of partitioning elements in the DNA to regulate the genome. And so I'd like to acknowledge this work was done during my PhD thesis at UCLA in the laboratory of Guillaume Chanfreau, and had a lot of help with these projects from my fellow grad students, Jason, and some technicians and undergraduates as well. And I'd like to thank the Lexogen company for developing the QuantSeq library prep method and for troubleshooting the protocol optimization. And we performed the direct RNA sequencing with the Helicose platform with seq-LL. And now I'd also like to thank the funding sources, and thank you very much for your attention.
Thank you, Kevin, for your presentation. A quick reminder for our audience on how to submit any questions they might have for you. You can submit your questions simply by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And Dr. Roy will answer as many of your questions as time permits today. I see that we do have a question in our queue. Dr. Roy, an attendee asks, why would one want to perform 3' end sequencing instead of full-length transcript sequencing?
Thank you, Brenda. So that's a good question. And if we look back at the full-length transcript sequencing, we can see that a large number of reads are required to map the transcripts. And so with a genome that's not well characterized, the full-length transcript sequencing can be important to first identify where the genes are. But with genomes that have been previously mapped, the 3' end sequencing provides a superior approach for quantifying gene expression and for mapping the precise ends of the transcript.
I see. Okay, we do have another question. How many roadblocks for RNA polymerase were identified in your studies genome-wide?
So using the nuclear exosome targeted poly-A tails as a signature of these roadblocks, we found on the order of 700 roadblocks genome-wide if we consider a cutoff of 1 read per million. And so these roadblocks were found upstream of these Rrb1 DNA binding sites as well as the TF3B binding sites. In addition to those two factors, other DNA binding proteins were also identified in our study.
Okay, great. Our next question. Are the DNA binding roadblocks you found all 9 BP in gap?
So the 9 base pair gap that we identified, if I go back to this slide here, that is that is the gap between where the DNA replication is roadblocked and the RNA polymerase is roadblocked. And so what's actually happening here is, if you look at the bottom left panel, the RNA polymerase is coming in from left to right on the plus strand, and the black bar at the bottom represents the protein binding site. And so what we conclude here is that RNA polymerase is actually physically hitting the TF3B protein, and that the distance between the leading edge of the RNA polymerase and the active site of RNA polymerase is roughly 16 base pairs in this case. And so DNA replication, what we can conclude is the DNA polymerase has a shorter distance from the active site. So perhaps this DNA polymerase is slightly smaller, and so it can get closer to the roadblock.
I see. Thank you. We have a next question. I think it comes in two parts. First, thank you for the talk. And they ask, what alignment tool do you use for their first part of the question?
Okay, so we use the RNA star alignment tool, and this is great for mapping RNA, and it's also recommended by Lexagen.
Okay, great. And their second part of the question, do you think techniques you use to reduce the number of reads with multiple hits can be applied to DNA sequencing?
So the reads with multiple hits, these, in our case, these are the polyA tails that are mapping to the ribosomal RNAs largely, as well as other RNAs. And so this is really a specific issue for RNA sequencing. And so I don't think the techniques that we use here are going to apply to mapping DNA reads because they're specific to the polyA tail, which is not a part of the DNA sequence.
Okay. We have another question coming in. How much reliable 3' end sequencing is there in terms of RNA quantification?
How much reliable 3' end sequencing is there in terms of RNA quantification? I'm not exactly sure what the question is saying. In terms of quantifying gene expression, the previous webinar I think went into detail on this, and you can obtain reliable fold changes in expression by quantifying the 3' ends. For our studies, when we're looking at the nuclear exosome targeted polyA tails, we're actually seeing a very substantial change in level. So we actually use a cutoff of 5-fold to define something as targeted by the nuclear exosome. So it's a very stringent cutoff. So if you want more subtle changes, so such as 2-fold or less, you will need to have multiple replicates and utilize some statistics to determine that the changes you're seeing are significant. So we did not need to employ that kind of statistics in this study because we set such a stringent cutoff.
I see. Okay. I think our next question coming in, how did you deal with rRNA contamination in the in vitro poly-adenylated samples?
That's a great question. So I skipped over that detail. So actually, the first time that we did it, we got nearly 80% of the reads mapping to the 3' end of ribosomal RNA, and that actually was, it's really a waste of a lot of sequencing reads. And so the second time that I did this, we used the ribosomal RNA depletion kit after the in vitro polyadenylation, because these ribosomal RNA depletion kits they typically don't give you much yield, so we wanted to first perform in vitro polyadenylation with a substantial amount of RNA, let's say 10 micrograms or 20 micrograms, and then re-quantitate the RNA and then subject it to a ribosomal RNA depletion. In this case, we used the epicenter kit for that, which has these oligonucleotides that bind to ribosomal RNA to remove them from the sample. And that actually worked out quite well. I mean, after that, we reduced the ribosomal RNA reads to less than 5%.
Okay. Second part of that question is a bit of a follow-up. In these samples, were there a lot of repeated poly-A reads?
Were there a lot of repeated poly? So I think that question is perhaps asking if the oligo-dt was somehow internally priming within the poly-A tail, and that would lead to a substantial amount of poly-T, actually, because it's reading the poly-A tail during the sequencing reaction. It will read a stretch of T's. So we actually didn't see any increased prevalence of that with in vitro polyadenylation versus non-in vitro polyadenylation. And that is mitigated by the fact that the oligo-dt primer, I didn't indicate it in the slide here, but the oligo-dt primer actually has a V at the end, so a V meaning anything but T is at the very 3' nucleotide, and that actually substantially prevents the internal priming of the poly-A tail. And so we think that's an effective way to reduce this poly-T reads that are occurring.
I see. Okay, we have another question. An attendee asks, I may have missed this, but I was wondering, did you also look at if quant sequencing can identify gene expression level precisely?
Yes, it can. And in order to do that, again, you need multiple replicates, and you need to employ some statistical analysis on these replicates. But yes, it can. We didn't need to do that in this study, as I mentioned previously, but it's very effective, and I think the Lexogen website has some software recommended for doing that. That can be done with either the DE-Seq software, or I even believe there's a new software they have placed on the website.
Okay, a follow-up to that question.
Okay.
Did you have to clip off the first several base pairs or the poly-A tails of the reads?
So before mapping these reads to the genome, we clipped off the adapters that are sometimes present at the 5' end. So if you look at the slide here, the portion of the DNA here in blue is the adapter. And so occasionally the insert size is less than the read length. So if the insert size, let's say, is 40, the read length is 50, we're going to read 10 base pairs into the blue. And we want to clip that off. So we did that. At the other end, we did not need to do any clipping, mainly because the oligo-dt was efficiently hybridizing to the junction between the poly-A tail and the genomically encoded RNA. So we didn't have to do anything there.
All right. Lots of great questions coming in from our attendees today. Compared to full-length RNA sequencing, how much less sequencing was performed for 3'?
So we have done RNA full-length sequencing as well. And so the amount of sequencing depth that you want, it really depends on what transcripts you want to detect. So if you look here, this is actually a very abundant transcript. It's this RPL9B. It's a ribosomal protein gene. And so for these samples, for RNA-Seq, we we would obtain on the order of 10 to 20 million reads, which is good for yeast. Whereas for QuantSeq, to get an equivalent quantitative level of the transcript, you would probably only need a fifth of that, so say 2 million to 5 million reads. So it is more efficient in sequencing depth by maybe a factor of 5.
Okay. Another attendee asks, what did you use for your sample prep?
So I'm not sure exactly which part of the sample prep they're talking about, but initially we purify the RNA from the cells with a phenylchloroform extraction, and then after RNA purification, we use the QuantSeq library prep kit. Part of the RNA sample prep does involve a DNase treatment to get rid of the genomic DNA prior to that. And we do a limited treatment for 30 minutes at 37 degrees, and then we purify the RNA, and then we use the QuantSeq library prep kit directly.
Okay. Another question, how often is roadblock used for fail-safe termination of microRNAs, cuts, and SNO RNAs? And do you do any statistics on that matter?
Did you do any statistics on that matter? So we identified roadblocks at a large fraction of the sites. So we look here at the summary figure here. We can see that the most of the nuclear exosome targeted transcripts do not have this DNA resistant site directly downstream. However, what we found is that a large number of these were actually roadblock sites, but that the DNA binding proteins just were not mapped in a previous study to have a DNase resistant site there. So in total, we identified, depending on the cutoff, between 700 to upwards of 1,000 sites of roadblocking. And the roadblocking appears to be primarily used for these cryptic transcripts, so these cuts and the sets and zets. The distinction between them is not always clear, but less so used for mRNAs. But we do find that often that downstream of mRNAs, we also find roadblocking sites as well, but predominantly the cryptic transcripts are the major class.
I see. We have another question. How do you deal with internal priming?
Internal priming. So perhaps later they can go through that section again in detail, but the internal priming we dealt with first by raising the temperature of the reverse transcription to 42 degrees. That reduced but did not eliminate internal priming, and so we added the computational filter. Again, that's on slide 12 here, so I don't want to go over all that again, but we did these 3 steps to get rid of internal priming after the fact computationally.
I see. It looks like that is all the time that we have for our questions. We've had great participation from today's audience. I know we appreciate it. Kevin, do you have any final comments or thoughts for today's attendees?
Yeah, I would like to thank everyone for attending the talk, and I would be happy to answer further questions by email or through the Labroots mechanism that you have set up. And I'd like to thank Lexogen in particular for all the technical support along with the kits that they provide and for continually developing new products. And I'd also like to thank everyone that helped in this project. So thank you very much.
Okay. Well, thank you, Kevin, for taking the time to be with us today. I want to thank our audience for joining us and participating so much and for their interesting questions. And of course, thank our sponsor, Lexogen, for underwriting today's educational webcast. This webcast can be viewed on demand through December of 2016. Labroots will be alerting everyone via email when it's available for replay. and we would encourage you to share that email with any colleagues who may have missed today's live event. Until next time, goodbye.
Goodbye.